So
Yes,
sorry
thank
about
you.
this. Yeah, I had to cope with that noise. So where are we today? What would be the next kind of best model how to cope with the cancer biology? Where are we today? So I want to today present you a printer and the ductal carcinoma breast cancer model that we have, and it has been working very fine. So with printer, one can basically print any kind of different viscosity of different materials and combine them. So with printer, you can have multiple different kind of tool heads, and from all the way from liquids to kind of silicones, and always up to ceramic pastes and melted plastic. You can combine these into one object, and this allows us to make real models of what we have in our human bodies. So we can add everything into the same structure regardless of the stiffness of the material. So it can be low stiffness. For example, if we look at our brain tissue, fat tissue, or skeletal muscle tissue, or bone tissue, whatever. So we can add these into the same structure. And we are lucky now that we have these materials available together with the different cell lines. Here is one example that what we have done with printer. This is basically all about how the breast cancer develops in human. So we have ducts and lobules shown here in the top part of the picture, and the cancer usually only happens, or the worst kind of cancer that you cannot treat happens always in the ducts. So why does it happen there? That is a good question, and that this is something that we have been studying, for example. And this kind of questioning requires 3D printing. Otherwise, we cannot mimic the same situation that we have on a plastic. So we need to 3D print the same kind of environment that we have on a human breast. Otherwise, it's the results would be non-convenient. So we have the nipple, we have the ductal part, and then we have the lobules that produce the milk. And then we have the stroma. So by the stroma, I mean that we have this kind of tissue that has different kind of structures inside of it. And the cells will always interact with the structure, they will always interact with the fibers, etc. So, for example, in this case, we have taken different kind of imaging to cope with this. So we have mouse breast tissue shown here on the upper left corner, mouse breast tissue, and then we have a human breast tissue as well. And then we have this kind of imaging sectioning that we can mimic this structure and how to replicate this one. And this is very important. So now we are at the stage with this cancer studies where we have the ability to image and mimic and replicate the tissue. And it's very well known that we also know the kinetics So we can now combine all the different layers of the data that is out there. So replication of this is very important. So for example, here we simply took the image data and replicated this and printed this. And the idea behind this study is to get the cell lines in their correct format replicated by robot, so kind of the 3D printer, and see how well they cope and how well they interact with the fibers produced. So for example, here we designed simple design model, replicated that with a 3D printer and see what is the reaction of the cell lines when printed. Based on the model, for example, here in the middle, you can see that the cell lines grow very well. We can actually modulate the matrix, so we can have the same stiffness that the breast cancer has. So it's always a stiffer structure when we are talking about cancer. So we can modulate that. And in this case, we were happy to see that the printed cells were very viable. They are actively growing different kind of pheroids. And what we can look at this study is that we have this invading cell lines going out of the pheroids as well. So that is the most important stuff. So in the end, by doing this 3D printing, we can actually look what are the forces that the cell lines make, or the interactions that the cell lines make with the matrix. And this is exactly what the kind of cell invasion is all about. If the cell cannot attach to its surroundings, it cannot invade, it does not make metastasis. But here, first time, we can take a look of how well the cells interact with their surroundings and how are they invading to their surroundings. And on the left upper corner, you can see this atomic force microscope image from the same printout, where we can actually measure the forces, how the cells invade to their surroundings. And kind of peaking these cancer models in more broadly scale, with 3D printing and with printer, we can sum up the dimensionality, stiffness, topography, at their different kind of growth factors, after different kind of interventions, drug, for example, and different kind of spatial signals. Past days, we were only limited by the factor that we needed to use cell lines that we purchased from different companies. But nowadays, we can use also patient-derived material. So that brings us to the fact that we can use 3D printing technologies towards personalized medicine care as well. And from my point of view, this is kind of the best way to use 3D printing technology in the future, especially when talking about cancer. So kind of we could use patient-derived organoids or patient-derived material to test if some kind of drug care is useful or not useful. So I talked about this printer's advantage already before. So we are not limited by any factor of the viscosity. We can use all the ranges, not only pneumatic tools, but we have also screw-driven tools. And I have to say that this Purdine, who is also sponsoring this meeting, I love that tool. It's outstanding. It's very good. I'm looking forward to kind of print different cancer models with that tool head in the future as well. One other thing that I would like to talk to you about is how to make this 3D printing to the next level. And it's about organs on chip. So past days we had 3D cell culture, animal models, spheroid models, but now we are able to do these organ on chip models that can be easily done if they are designed correctly, and there we can add and use them also in different kind of manners. So I mean, we can add their pulsating, for example, blood flow. We can use it, for example, COVID studies, so we can have their air-liquid interface, etc. And if we add there also the capabilities of the 3D printing, so that would open totally new kind of scenarios for different kind of material studies, drug development, drug design, etc. So this is something to keep in mind, and I'm very forward looking in publications related to this area. And because we are now accessing different kind of materials, patient-derived materials, so there's no limitation of what we can design. So it can be not only cancer, but different kind of organs to be built and studied in the future. So by Printers Core team, I would like to thank you, Jenny and 3D Heals, for this possibility to say hello from Finland and to tell something about printer.
Thank you so much for your presentation. So our webinar typically would involve multiple continents in the time zone that sometimes somebody's going to be 5 a.m. or other people are going to be midnight. So I think, so I appreciate everybody who's participating and also speaking at these panels. So guys, if you have any questions, put them in the Q&A box, because I have tons of questions and I don't want to occupy the floor all the time. But I do have a couple of really quick questions, first of all, because I'm not a biologist, and you produced this nice map about the traction force. How do you measure these traction? What technology do you use to measure those?
So that is basically atomic force microscope. So in theory, there is simple like small beads just laying around in the matrix, and when the cells contract, they will pull these beads towards them.
Okay, cool.
And that is the simplest thing to measure kind of the forces. Also, the key theory in this is that you need to see the fibers. The fibers are very important how to kind of mimic the tissue structures. So collagen fibers, etc. So there's many different ways to kind of visualize them. For example, either you simply have fluorescent collagen fibers, or then you use these harmonic waves to measure these collagen fibers. So I guess those are the key technologies in there.
Yeah, cool. That's awesome. I mean, I never, you know, my own medical practice, I don't usually think in a microscopic stage where you really look beyond what you can see with your eyes or on CT or MRI. And this is the first time I actually really think from a cellular level. And the way that you described how breast cancer developed is very insightful for me. So I'm just curious, I'm sure that you guys are also looking into other really common cancer lines, like lung cancer is one of the top killer right now. And the other one is melanoma. That's affects all age group. Have you thought about these other kind of cancer? Do they also have as much insight as we do for breast cancer?
Yeah, so that is kind of the most common question that I get. So what kind of cancer are you studying? So based from a 3D printing and technology point of view, it doesn't matter what kind of cancer you are studying as long as the model is correct. So if you get the good kind of cell line that mimics the cancer that you are studying, it doesn't matter. As long as you can print the correct kind of model, so then it's good. So colon cancer or breast cancer, from the technological point of view, yeah, of course there's limitations. Of course there's different kind of drugs that the doctor can give you or the patient in the end, but from a printing point of view, that's not the limiting factor. We can kind of print all different kind of models of cancer.
Awesome. Thank you so much. Okay, we're going to move on to the next speaker. And for those guys, those of you who have questions for NT, please put your question in the QA box, and we're going to address all the questions after all the panelists present. So thank you so much. So the next speaker is going to be Dr. Carolina Valente. She is currently faculty of University of Victoria, correct? But also a CEO and co-founder of a startup called Voxel Innovation. And her main subject is oncology. So there's no further, just that's her focus. So I'm pretty excited to hear about what you're working on, Carolina.
Thank you, Jenny. Thank you for the kind introduction. Can you see my screen well?
Yeah, perfect.
Yeah, thank you. So as Jenny said, I'm here to talk a little bit more about bioprinting cancer, and I'm providing a little bit of background of what happens in cancer and why it's so hard to mimic cancer in these preclinical testing techniques that we're currently using. And I will finalize talking a little bit about how we are tackling these problems with Voxel and what we have been doing in terms of our cancer tissue models. So cancer is a devastating disease. We know that for a long time. In terms of, we are currently located in Canada. So in terms of the Canadian perspective, is 50% of the population will get cancer at some point, in which 20% will die from cancer. And last year alone, in Canada was close to 85,000 people dying. And of course, if we put that in the US perspective, it's even larger. The number is even bigger. So we refer to cancer as a single word, cancer, but what we know is multiple different diseases under that cancer umbrella. And because there are multiple different diseases, we can treat in different ways. So cancer treatments, they revolve around chemotherapy, radiation therapy, and hormonal therapy. The challenge with these therapies is what we see is, even for example, for breast cancer, that has been one of the topics today, is the two different people can have breast cancer and they will react differently. And that shows you the complexity of the breast cancer or the cancer in general, the cancer in microenvironment.